Students, now let us discuss some of the competitive bits related to the topics molecular diagnosis and transgenic animals. Question number 1. Early detection of disease is possible by option A, gene therapy, option B, recombinant DNA technology and ELISA, option C, PCR, option D, both B and C. That is, early detection of disease is possible by. So you should be able to know the question is coming from which concept, and you should be sure of the answer. You should know what is the specificity of each of the option given. Then only you will be able to solve these need bits. So this is one of the trick. How you'll get to know the specificity? When you keep on reading the textbook. How many times you will read the textbook, that much it is going to be beneficial when you are writing the competitive bits. Okay, so option A, gene therapy. Gene therapy is a technique that is used to treat the disease. It is used to treat the disease, not for the diagnosis purpose. Right? Then option B, recombinant DNA technology and ELISA. Yes, ELISA is enzyme-linked immunosorbent assay. It is based on antigen and antibody interaction. It is also used to detect the disease at the early stage. Either the antigen is invaded in the body, or if there is a first thing the body is going to respond to the antigen is by production of antibodies. So either of it can be used for the testing purpose. So ELISA can be used for detecting disease at early stage. Option C, PCR reaction, that is polymerase chain reaction. Okay, yes, polymerase chain reaction is going to amplify the DNA in the early stages of the disease. Okay, the genetic material that is infected can be amplified. The low concentration of the genetic material can be amplified using PCR reaction, and then the detection of the disease at the early stage can be identified. So option D, both B and C. Yes, option D, D for dog, that is last option, both B and C, that is recombinant DNA technology and ELISA and PCR. These two are the techniques that are used for early detection of disease or early diagnosis of a disease which is required for efficient treatment. So option D is the right answer for question number 1. Question number 2. Technique used to detect the DNA in clone of cells is. The technique used to detect the DNA in clone of cells is option A, PCR, option B, gel electrophoresis, option C, chromatography, option D, autoradiography. There is a spelling mistake here. The GR is PHY. No, it is autoradiography, GRAPHY. Okay, the technique used to detect the DNA in clone of cells. PCR, no, it is going to amplify the DNA. So, and gel electrophoresis, option B, it is used for separation and isolation of the DNA fragments. So, it is a separation technique. And option C, chromatography, is going to purify the sample. It is going to separate the two closely related substances, and hence it is a purification technique. Autoradiography, yes, here the radioactively labeled elements are used which are tagged to the single stranded DNA or RNA, and these probes are allowed to hybridize with their complementary DNA sequences in the clone of cells. So option D, autoradiography, is the right option. That is, the technique used to detect DNA in clone of cells is autoradiography. So option D is the right answer. Moving on to question number 3. Single-stranded DNA or RNA tagged with radioactive molecule and is used to detect mutated genes is called single-stranded DNA or RNA tagged with radioactive molecule and is used to detect mutated genes is called RNA interference. Option A, option B probe, option C plasmid, option D primer. So just now I told these probes are radioactively labeled molecules. Okay, they are tagged with the single stranded DNA or RNA. So option B probe is the right answer for this question. If you see other options, option A RNA interference, it is a production of transgenic plants, that is to produce a plant resistant to parasite nematode. The technique used to produce the transgenic plant is RNA interference. It is not a probe. So option C plasmid. Plasmids are extra chromosomal double stranded DNA. They are used as vectors to carry the genes from one organism to another organism, and these are once again they are not the probes and they are not used to detect the mutated genes. Option D primers. Primers are used in PCR reaction. So the right answer for this question is option B probes is the right answer. Question number 4. Pathophysiology is the study of physiology of pathogens, study of normal physiology of host, study of altered physiology of host. Option D, none of the above. Pathophysiology is option A, study of physiology of pathogens. No, the study of physiology of pathogens is also called as physiology only, not the pathophysiology. Option B, study of normal physiology of host. It is physiology only once again. Option C, study of altered physiology of host. Yes. Option C is the right answer. It is a study of altered physiology of host. When there is alteration of physiology is going to take place, when there is a abnormal functioning of the host is going to take place, when there is a invasion of foreign body into the host cell, then the physiology is going to be altered, which is obtained by the invasion of the externally invaded or a foreign body, or it could be a pathogen. So it is referred as pathophysiology. The change in the physiology or the alteration of the physiology due to the presence of the pathogen or due to the invasion of the foreign body is known as pathophysiology. Option C, study of altered physiology of host, is known as pathophysiology. Option C is the right answer. Question number 5. A molecular diagnostic technique based on antigen antibody reaction is the technique that is used for detecting purpose or for diagnosis purpose, which is based on antigen antibody reaction, is option A PCR reaction, option B DNA hybridization, option C ELISA, and option D serum analysis. The molecular diagnosis where you can detect a disease at molecular level. So anyways, PCR is also a molecular diagnosis method, but here the amplification of gene of interest is going to take place, means you are going to amplify low concentration of DNA to billion copies, which is useful for the studying purpose. And here the primers are involved, antigen and antibody is not involved. Option B DNA hybridization, here once again the probes are involved which are tagged to the radioactive molecules, but antigen and antibody is not involved. Option C ELISA, yes, it is an enzyme linked immunosorbent assay wherein antigens and antibodies are assayed or they are detected for finding out the root cause for the disease, and it is based upon, the process is based upon antigen and antibody reaction, right? if either the antigen or antibody is immobilized on a solid microplate and externally its counterparts are externally added onto it, okay, and allowed to react with this antigen or antibody, which are in turn linked to the reporter enzyme or a secondary antibody, which is on reaction with the substrate is going to produce a color change. Based on this, the amount of infection or the presence of the infection can be found out. Option D, serum analysis. Here the biomacromolecular assays can be done, but not the antigen antibody reaction is involved. So option C, ELISA, is a right answer for this question. Question number 6. A technique used to detect AIDS in suspected patients in very early stages is. A technique used to detect AIDS in suspected patients in very early stage. In the early diagnosis, there are two methods. One is PCR reaction and one more is ELISA technique. So let us see which technique is used to detect AIDS in the suspected patients, and that are at early stages. Option A, PCR reaction. Option B, autoradiography. Option C, ELISA. Option D, DNA hybridization. Okay, PCR reaction, yes, it is used for detecting the mutated gene in cancer patients as well as for the suspected AIDS patient, right? So option A, PCR, is the right answer. Option A, PCR, is the right answer. That is going to amplify the low concentration of the DNA, wherein it is going to measure the DNA that is going to produce the antigen known as P24 antigen. So option A, PCR, is the right answer. Let us see autoradiography. The PCR is followed by autoradiography in case of suspected cancer patients to detect the mutated gene, but it is not used to detect the AIDS suspected patients. ELISA can be used for the AIDS patient who are positive, AIDS positive patient, as well as the qualitative also can be done, but it is used for quantitative analysis in case of AIDS. And DNA hybridization is, once again, it is not a technique to detect the AIDS in suspected patients. So option A, PCR, is the right answer. Question number 7. In ELISA, the end of the reaction is accomplished by option A, antigen, option B, antibody, option C, reporter enzyme reaction with its substrate to produce color, option D, none of the above. Okay, in the ELISA technique, if you see the step-by-step reaction, okay, first the antigen or antibodies are immobilized on a microplate and then their counterparts are allowed to react. If an antigen is immobilized, externally antibodies are allowed to react with it, or if the antibodies are immobilized, the antigens are allowed to react with it. Right? Then the washing step will take place. The unbound antigens or antibodies will be washed away. Okay, and the external counterparts which are allowed to react with the immobilized reactants are in turn linked to the enzyme known as reporter enzyme. They are known as reporter enzyme, which is going to it is going to bind to the antibody and form a antigen antibody complex will be formed, which is going to react with a particular substrate and it is going to produce the color. So based on the color change, okay, in the ELISA, the reaction is reported or concluded. So that is the reason that enzyme is known as reporter enzyme. And option D, none of the above. So the end of the reaction is accomplished by reporter enzyme. So option C is the right answer. Sometimes the question might be a bit twisted. Instead of reporter enzyme, there could also give the secondary antibodies. Secondary antibodies will react with another type of these antibodies only, and which is not going to react with the antigen, but it is going to react with the primary antibody, and it can once again result in the detection or completion of the reaction. So option C is the right option. Question number 8. Animals that have had their DNA manipulated to possess and express a foreign gene are called. Animals that have had their DNA manipulated to possess and express a foreign gene are called transgenic animals, hybrid animals, super animals, dual nature animals. What are transgenic animals? Transgenic animals are those animals whose DNA is manipulated or altered to possess a foreign gene or express a foreign product is known as transgenic animal. So option A, transgenic animals, is the right option for this question. Let us move on to the next question, that is question number 9. 95% of the existing transgenic animals are option A fish, option B pigs, option C sheep, option D mice. So there are transgenic animals or the models created for all type of these animals. There are transgenic fishes, transgenic pigs, transgenic sheep and transgenic mice. Among all these, the largest or the highest percentage of the transgenic animals present is option D, mice. The transgenic mice are the large variety of the transgenic animals present over here, because the mice have the tissues and organs that are similar to that of the human beings, and they can express the genes that are expressed in the human body. That is the reason the transgenic mice is preferred for the various applications related to human beings. So option D, mice, is the right option. Moving on to question number 10. Transgenic animals are created not to study which of the following. Transgenic animals are created not to study which of the following. Option A, regulation of genes. Option B, study of diseases. Option C, for biological products. Option D, diagnostics. Okay, let us see each option now. What are the advantages of the transgenic animals? Okay, but the question is, why the transgenic, for which purpose the transgenic animals are created not to study. Option A, regulation of genes. Yes, the transgenic animals are created to study normal physiology and development. Okay, so therefore it helps in to study the regulation of genes. So option A is a one of the advantage of transgenic animal only. Option B, study of diseases. Yes, the transgenic animals are created to study various diseases. What are the genes responsible in the causing a disease? Okay, based on this, new treatments are discovered. By knowing the root cause of the disease, the gene is coding for what type of product. Based on that, the genes are expressed to form those products to produce the new drugs or new treatment, right? So this is also one of the advantage of transgenic animals only. Option C, for biological products. Yes, we have seen in today's class, the transgenic animals are used to produce biological products, such as products that are derived from the animal cells, or it could be the components of these cells itself could be used as a biological product. For example, the production of alpha 1 antitrypsin and alpha lactalbumin. These are the two proteins. One is having the medicinal value to treat the emphysema, that is alpha 1 antitrypsin, whereas the alpha lactalbumin is a protein rich milk. So they are used for producing biological products. Option D, diagnostics. Okay, we have not studied the usage of transgenic animals to diagnose a disease, to find out or detect the disease. So therefore they are not created to study diagnosis of the disease, but is to produce certain products that are useful for medicinal value, or that is required in the day to day life. So option D is not related to advantage of the transgenic animals. So option D is the right answer for this question. Question number 11. Transgenic models do not exist for rhinitis, cystic fibrosis, rheumatoid arthritis, Alzheimer's. Transgenic models do not exist for rhinitis. Rhinitis is the inflammation of mucous membrane in the nose. Okay, rhinitis is the inflammation of mucous membrane in nose, leading to the swelling of mucous membrane will lead to the stuffiness as well as the running nose. So it is one of the disorder that is produced due to the common cold. Okay, so option A is rhinitis. Option B, cystic fibrosis. It is produced due to the non-functional gene coding for dysfunctional protein, CFTR protein. Cystic fibrosis transmembrane regulator gene codes for the CFTR protein. Okay, this is used for maintaining the chloride ion channel as well as the transport of fluids in the cells. The dysfunctional gene for CFTR results in the accumulation of mucus, sticky mucus, that results in the related complications. And rheumatoid arthritis, it is a joint pains, okay, and it is autoimmune disorder. Alzheimer's, it is a loss of memory. Okay, the transgenic models are used to study option B, option C, option D, but there are no transgenic models to study rhinitis, because rhinitis is not discussed in the textbook, or there is no any proof that is telling that the study of rhinitis is done in transgenic models. So option A, rhinitis, is the right answer. Question number 12. Insulin like growth factors are involved in growth. This study can be done by. Insulin like growth factors are involved in growth. This study can be done by option A, by introducing genes from other species that cause formation of this factor, by introducing genes from other species, by introducing genes from other species that alter formation of this factor, and option C, by studying genes from same transgenic animal which cause correct formation of this factor, and option D, by studying genes from other species which causes correction in growth pathway. Insulin like growth factors are involved in growth. This study can be done by, okay, that is by introducing genes from other species that alter the formation of this factor. Now how we are going to study the insulin like growth factor? We are inserting certain genes that is going to alter this growth factor, either it is overexpressed or it is less expressed, and their effects are studied. So the option by introducing genes from other species that alter formation of this factor is a right option. I forgot to type it here, but anyways the notes which I am forwarding to you people will contain the right options for question number 12, that is option B, by introducing genes from other species that causes alteration of the factor that is expressed in the transgenic animals. So option B is the right answer for question number 12. Moving on to question number 13. Advantage of transgenic animals in study of disease is. Advantage of transgenic animals in study of disease is to investigate new treatments for disease, to increase the Indian economy, possibility of early detection can be investigated, possibility of preventive measures can be investigated. Advantage of transgenic animals in the study of disease. One is we are expressing the genes that is going to cause the disease in the transgenic models. Why we are creating these models? To produce a particular type of disease, to know how these genes are contributing in the uh, production of the disease. What are the components that are coded by these genes that is resulting in the formation or resulting in the abnormal functioning of the biological process. So if you know the pathway or the route by which the disease is caused, similarly the treatments can be discovered to prevent this particular disease or to treat this disease. So option A, to investigate new treatments for diseases, is the advantage of transgenic animals in the study of disease. So option A is the right answer. Option A, to investigate new treatments for disease, is the right answer. Not to increase the Indian economy, or the early detection of the disease is also not possible using the transgenic animals. Okay, and possibility of preventive measures can be investigated, that can be done, but the main thing is we can investigate new treatments for the disease. You can create the disease condition in the transgenic animal. You can try for the different drugs which is going to cure that particular disease. So option A is the right answer for this question. Moving on to question number 14. Transgenic animals are those which have option A, foreign DNA in all its cells, foreign RNA in all its cells, foreign DNA in some of its cells, and option D, both A and C. Transgenic animals are those which is going to possess the manipulated or altered genomic DNA. Means, what do you mean by genomic DNA? The genetic material is going to remain same in all the cells. So, it should contain the foreign DNA in all its cells, not the foreign RNA in all the cells, and not the foreign DNA in some of the cells. Right? It must possess foreign DNA in all its cells only, not the foreign RNA and not the foreign DNA in some cells. No, that is the reason if we are inserting the gene of interest in the embryonic stages, so when these embryonic stage cells, when they undergo repeated cell division and they are getting differentiated, so as the it is going to produce different cells, tissues and organs, each will possess this manipulated DNA. So that is the reason it is going to form a transgenic animal. So option A, foreign DNA, foreign DNA in all its cells, is the right option. So question number 14, option A is the right answer. Foreign DNA in all its cells is the right option. Moving on to last question, question number 15. PCR will allow to detect genetic defects but doesn't involve which of the following steps? It will allow to detect genetic defect but does not involve which of the following steps? Probing with radioactive molecule, hybridization, autoradiography, antigen antibody interaction. PCR will allow to detect genetic defects, but which step it will not involve? Probing with radioactive molecules, yes, the primers can be used that are linked to the radioactive molecules. Hybridization, whatever the PCR products are there, it is denatured using restriction endonucleases, and they are allowed to hybridize with their complementary parts which are single stranded DNA or RNA linked to radioactive molecule. Then autoradiography, whatever the fragments obtained from the PCR, okay, and attached to the radioactive molecules can be detected on the X-ray film in autoradiography. Option D, antigen antibody interaction, is one of the main principle behind ELISA technique, not the PCR reaction. So the step which is not involved in PCR reaction during the detecting of genetic defects or the mutated gene in cancer patient is antigen antibody interaction. It is not involved. So option D is the right answer. So next, the last topic left out is ethical issues related to biotechnology. We will take up it in the next class. That is going to complete your chapter Biotechnology and Its Applications. Please go through the textbook and the notes what I am sending. If you have any doubts, you can please clarify it, because it is entirely a new topic to your people. So once you read and once you follow the notes and textbook, then it will be easy for you people to understand. Thank you.